Hello everyone. As we have already discussed about fertilization, its types, and in mechanism, we are going through with the first, that is approximation of gametes, and later next we'll see about its capacitation and contact, how sperm will help in contact of egg and what are the reaction that takes place further contact. Okay, so what is this capacitation? So we know like the sperm structure. So usually in mammals there will be no filament will be formed during the acrosomal reaction. So we know already acrosome, that is the a type of or structure which is present in the head region of the sperm, so which contains enzyme for rupturing of egg membrane. So now what happens, this during formation of acrosomal reaction there are some lytic enzymes. During the acrosomal reaction in mammals there is alteration in sperm morphology that allows to penetrate the egg. So what are the changes that happens? The sperm that leaves the testis proper. So you know, you have already studied the structure of testis. So during that time, what are the changes? So there are two types of maturation steps. First maturation that occurs in tubules of the epididymis, and the second maturation that takes place within the female reproductive tract. So that is called as capacitation. So you should remember there are two steps. First maturation takes place in tubules of epididymis. Second maturation it takes place in the female reproductive tract. So this female reproductive tract maturation, that is called as the capacitation. So there are some techniques you can alter the capacitation also. So capacitation may result in the form of removal of some inhibiting material from the sperm. So one of the usual technique that takes place in mammalian sperm. So they are using Sandai virus, so which helps in agglutination of the cells. So usually the head region of the sperm, they come in contact with the other sperm head, then what happens, agglutination takes place. Usually this Sandai virus is commonly used to bring about fusion of membranes of two cells. So it may be a acrosomal membrane or the sperm membrane. So this is about the capacitation. So remember two maturation steps and little about how this capacitation can be inhibited by using Sandai virus. Next we'll discuss about the acrosomal reaction and its penetration. So we already know that the acrosome, it is present in the head region of the sperm and it consists of some proteolytic enzyme. So once they are exposed, then only the reaction takes place. So what usually happens during this acrosomal reaction? So we know that for fertilization there should be a mutual interaction between sperm and egg. The intact sperm, it emerges from the male and it is not capable of penetrating egg. So what happens now? Firstly, it must undergo acrosomal reaction in response to egg environment. Once it comes in contact with egg, then only the acrosomal reaction takes place. So what are the series of changes that happens then? So as you can see in the diagram, there is release of enzymes, the anterior sperm plasma lemma, which consists of inner acrosomal membrane. So there are two regions, one is acrosomal vesicle and acrosomal granules. So we'll see in detail. So acrosomal reaction that begins with the sperm tip contact with the outer egg envelope. So first what happens? The sperm tip has to come with the contact with egg envelope. The first response, there are two membranes as I said, the outer cell membrane of the sperm head and the anterior membrane of acrosomal vesicle. So granules are the small dot like structure, those are the granules. Vesicle is the space between the nucleus as well as the acrosomal vesicle. So these membranes are always continuous and they never found in the free end. So next what happens? So usually this membrane has to split transversely. So once it split transversely, it appears to undergo immediate fusion. So what type of fusion it takes place? So the fusion of membrane, they are generally a simple type. That is how it takes place by the secretion. Once the fusion occurs, the contact of vesicles are continuous and through the external medium. So later what happens again? The membrane, they are derived from the Golgi vesicles. So as I said, there are two parts, vesicles and one more is granules. So these vesicles, they are formed by the Golgi vesicles, and in the case of the acrosome, they are particular to fuse with the outer cell membrane. So later one membrane fuse with the another, the egg membrane. So what it consists of? The lysine, it will be secreted in the posterior wall, and later the rod like projections. So once the egg is coming in contact, so this egg membrane will be ruptured and later rod like structures will be formed. So those projections, what they will do? So usually they grow up to in the length, and that is the first actual contact with the plasma membrane of the egg surface. Till now there was not any contact between sperm and egg. So once there is a contact, then the acrosomal reaction will start. So again, in this process what happens? The membrane fusion occurs, but the event involves the acrosomal membrane of the sperm and plasma membrane of the egg. So both sperm and the egg, now there is an exchange of the material. At this point we can tell that what we can tell, two gametes, that is sperm and egg, they are coming in contact with each other and they are forming a single cell, that is zygote. So later fusion, what happens then? So this event will be provided with the open channel in the interior wall of sperm head and male nucleus. So later the midpiece gains the entrance and slowly it will pass towards the egg cytoplasm. So to bring together egg spermatozoa, the spermatozoa are released on contact, penetrate the body wall and make their way through the body tissue to reach ovary. So this is how the fusion of sperm and egg takes place once the acrosome reaction will go on. So this is about the acrosomal reaction which takes place in between sperm and egg.